Thank you, everyone. So thrilled to be starting today's session. It's really exciting. So, I'd like you to come with me on a voyage from dusty old Israel and stinking hot Tel Aviv to one of my favorite corners of Australia. Not so far from where I grew up, the lush, volcanic, subtropical rainforests of southeast Queensland. Now that you're with me, let's go for a walk, as would say down under. Let's go bush bashing and forge our own path through the dense undergrowth. As we hack our way along the trails, crossing creeks and swimming in waterfalls and enjoying the birdsong amongst the trees, you start to feel something itching away at your ankle. Despite your best efforts to ignore it, you instinctively swat at the unpleasant sensation, and to your horror, find yourself face to face with a massive funnel web spider, fangs dripping with neurotoxic venom. Oh, terrifying. Thanks to your quick reflexes, you manage to do the natural thing and you yeet that spider deep into the bushes and live to walk another day. Thinking as a neuroscientist and as a less than empathetic friend, the most miraculous aspect of your near-death experience is not just your mere survival, but rather the fact that a faint sensation caused by an animal many, many times smaller than you traveled all the way up your leg, into your brain, and became a conscious and actionable thought within a fraction of a second. This miracle, the miracle of sensation, is made possible by some of the most unique cells in our body, our sensory nerve cells. So sensory neurons are actually the longest cells in the animal kingdom. Despite having a cell body a few thousandths of a millimeter in size, they send out cellular cables, known as axons, out to more than a meter in length in our longest nerves, like the sciatic nerve. These cells can also grow really rapidly, in some organisms up to centimeters a day as the animal develops. And unlike the neurons of the central nervous system, they can even regenerate after injury. However, just like the Spider-Man you very nearly became, with great growth comes great logistical challenges. For proper function, the cell body, which is kind of like the logistics hub of a cell, and the axon tips, which receive input from the outside world, must be in constant contact with the axon in between, acting like a thin, delicate wire. So, logistical issues. How are crucial cellular resources like proteins and RNA transported over such vast and enormous distances as the neuron grows? How do neurons integrate and respond to various signals and stimuli coming from their most distant regions, perhaps a chronic source of pain? And most relevant to today's talk, how do neurons plan their recovery if the fragile connection between cell body and axon has been severed? Think even more broadly, why do sensory neurons in the peripheral nervous system regrow after injury when central nervous system neurons can't? What genetic programs make this possible? So to answer these sort of questions, me and my lab use various genomic tools and animal models to study gene expression at the systems level. But I want to take today's opportunity to tell you about a recent and very unexpected story straight from the lab bench. So, we started with a model of sciatic nerve injury in mouse, which we followed up a few days later at the peak of the injury response and as nerve regrowth is just beginning by performing deep RNA sequencing on the sensory neurons themselves. This is a technology that allows us to essentially conduct a census of all of the RNAs being expressed in a cell at a given time. Actually, just as an aside, and as with every good science story, we were looking for a completely different phenomenon, something related to RNA processing, but I'll spare you the details because, of course, we found no evidence for that going on in these cells. However, as we started to pore over the apparently negative data, what we did find was a really strong and really specific increase in a very strange type of RNA known as B2 signs, depicted here in their folded state by this lovely blue squiggle. But who is this enigmatic character, and why do I keep saying that this is such an odd finding? To understand it a bit better, you'll have to indulge me and let me give you a brief, short, oversimplified genetics lesson. So, the mouse genome, and for that matter the human genome, is made up of 3 billion or so letters, our genetic code, our nuclear bases, that make up, basically what makes me, me, and you, or you, and you, me, within which we find the about 20,000 or so genes that become proteins. However, it might surprise some of you to know that this portion of our genome, the protein coding genome, only makes up about 2% of the genome as a whole. This remaining 98% puzzled a generation of geneticists who somewhat disparagingly referred to this genomic wilderness as the junk genome, full of useless DNAs and RNAs with no apparent function. Sign RNAs, which is actually a kind of soulless catch-all acronym for short interspersed nuclear elements, were too sadly lumped into the junk genome. But like flies around a trash can, they've actually multiplied throughout the genome over the course of evolution. There's actually more than about 500,000 sign sites scattered throughout the mouse genome. However, what gave us a clue that the sign RNAs that we were seeing after injury were real, that they were in fact signal and not noise, was the fact that of all of this multitude of sites, only a tiny handful, equivalent to 1/2 of 1% of one specific subtype of sign RNAs, those B2 signs, were being activated after injury. Even more encouraging was the fact that most of these sign loci that were being increased, upregulated, were under the control of one of the main transcription factors that we know to be active in the injured neuron, perhaps suggesting that B2 signs are part of a crucial regulatory switch that is suddenly turned on. But what biological function could this junk RNA possibly be performing? Are B2 signs merely a passenger in the genetic programs that drive the recovery from injury, or do they play an active role in shaping the injury response? It turns out that B2 signs are potent activators of nerve growth. If you block the expression of B2 sign RNAs in a culture of mouse sensory neurons, you almost completely abolish the outgrowth of long axons, as you can see in this microscopy image here, where you can see the stunted axons in cells that have been starved of B2 signs. So as I was telling you, while we found really robust increase of B2 signs in the peripheral nervous system, when we looked at injury models of the central nervous system, we saw actually no change in sign expression whatsoever. Nothing at all, potentially suggesting that these systems, as we know, are very, very different. So what happens if you introduce a B2 sign into the central nervous system where, as we know, the neurons don't recover after injury? What we saw was quite remarkable. When you introduce B2 sign RNAs into the central nervous system after injury, you have dramatically enhanced axon regrowth across the injury site, which is a vital first step towards functional recovery. So the air is now heavy with what I feel is the obvious question: can we harness the junk genome, the junk RNA of the human genome, to help regrow injured human nerves? This lofty goal is one of the key questions I hope to answer over the next few years. Thanks, Azrieli. Well, to begin with, we have to establish whether human neurons actually express their own version of some kind of injury-induced sign RNA. And we think they will, we expect they will, because most of the injury pathways discovered thus far tend to be shared from mouse to man. But there are some key differences that make this just a little bit more complicated. To begin with, in comparison to the genes that code for proteins, sign RNAs and signs in general are under much lower evolutionary pressure. And as we've diverged from other mammals, so too has their genetic profile drifted. We've developed our kind of own repertoire of very different sign families, our own sign language, if you'll pardon the pun. And exemplifying this is the fact that one of the main signs in the primate lineage, alloRNAs, comes from a completely distinct molecular heritage from the B2 sign RNAs found in rodents. You can see the squiggles are completely different, which is hard science. Even complicating things just a little bit further, signs have, for whatever reason, massively increased in abundance over the course of evolution in our genome. There's at least twice as many in our genome, meaning that we could end up looking for a needle in a haystack, which is a challenge, but not an insurmountable challenge, thankfully. Perhaps most tangibly, last I checked, there isn't a very long line of people waiting for us to perform somewhat invasive nerve injury recovery experiments, so we need to build some of our own models to help study human signs. So what we want to do is build a culture model that comes as close as feasibly possible to an intact human nerve. To this end, we're thankful to recent advances in the stem cell field, where what we want to do specifically is we want to use a genetically modified line of pluripotent stem cells, which can be directly differentiated into sensory neurons with efficiencies of essentially 100%. What's nice about these neurons, and as you'd hope for any good model of sensory biology, is that they respond to various sensory stimuli and form real interneuronal connections, allowing us to build truly physiological neuronal circuits. To make this model even more real, we are currently generating custom cell culture devices with nanopattern growth surfaces that cause the axons of these neurons to naturally bundle together and grow in a very well-arranged pattern out to extreme distances. When put all together, these tools will allow us to simulate axonal injury in the human neuron, study their recovery and regrowth, and hopefully, with some of the omics tools I described earlier, we can catch a whiff of some injury-induced junk RNA along the way. So, to sum up, I'm right at the beginning of this project, and it's very tempting to wildly speculate and wax lyrical about the therapeutic potential of human signs, yada, yada, yada. But what excites me most about this work is something much more fundamental, that a simple idea, followed by a simple set of experiments, led us to a place with just as many questions as answers. I hope that today's talk has been a good reminder that, just like that spider hiding behind that bush over there, the most exciting discoveries are often come from places where we least expect them. Thank you so much. I'd like to thank my lab.
Thank you, Adam.